Hello everyone, good morning. This is Bhargawi from class 11 from school Saraswati English Medium School Chandrapur. Today is our presentation of subject biology. Today we will explain you what is Gramstaining, steps of Gramstaining and difference between gram-positive bacteria and gram-negative bacteria. Now I will tell my friend to introduce herself.
Hello everyone, good morning. This is Jyoti from class 11th. Now I will explain you gram staining. To understand gram staining, first we need to understand that what is staining. So staining is a technique in which we can applying a coloring agent to the sample of microorganism from which we can observe the internal structure of microorganism by the help of microscope. Without using a stain, it is very difficult to visualize the sample of microorganism. Now I will focus on gram staining. Gram staining was originally devised by Hans Christian Gram in 1884. When bacterium gets stained, it is called gram staining. It is also called gram-positive bacteria and gram-negative bacteria. It is widely used in bacteriology. Now I will focus on its principles which are based on gram-positive bacteria. So first is gram-positive cells have more acidic protoplasm which may account for their retaining the basic primary dye more strongly than gram-negative bacteria. And the second is the peptidoglycan of gram-positive bacteria is thick and thus able to retain the diiodine complex. Now I will explain you steps of gram staining. So first step is fixation. Fixing a heat on a slide. Bacterium is heated to be fixed on a slide. And the second is crustal violet. The culture stained with primary stains which are crustal violet for 1 minute. And the third stage is washing the slide. And the fourth is iodine solution. Application of dilute of iodine solution which is also known as gram iodine. And at the fifth stage, bacterial color is purple. And at the sixth is decolorization. Decolorization of alcohol and acetone. And the last one is counter stain. Counter stain with carboluxing for 30 seconds. Now my friend will explain you the differences between gram-positive bacteria and gram-negative bacteria.
Now my topic is to explain what is difference between gram-positive bacteria and gram-negative bacteria. Before this, I will define what is gram-staining and gram-negative bacteria. Now I will explain difference between gram-positive bacteria and gram-negative bacteria. Before this, I will define the term gram-positive bacteria and the term gram-negative bacteria. First, the term gram-positive bacteria. Those retaining their color is called gram-positive bacteria and those doesn't retaining their color is called gram-negative bacteria. Now, I will explain difference between gram-positive bacteria and gram-negative bacteria. First, cell wall. In gram-positive bacteria, a single layered smooth cell wall is found, whereas in gram-negative bacteria, a double layered very cell wall is found. Second, cell wall thickness. In gram-positive bacteria, the thickness of cell wall is 20 to 80 nanometer, whereas in gram-negative bacteria, the thickness of cell wall is 8 to 10 nanometer. Now, peptidoglycan layer. In gram-positive bacteria, thick multi-layer peptidoglycan layer is found, but in gram-negative bacteria, thin single layer is found. Now, fourth, teichoic acid. In gram-positive bacteria, teichoic acid is present, whereas in gram-negative bacteria, teichoic acid is absent. Now, outer membrane. In gram-positive bacteria, outer membrane is absent. In gram-negative bacteria, outer membrane is present. Similarly, there are more differences between gram-positive bacteria and gram-negative bacteria. Now, porins. Porins is a hole. It helps to transformation of proteins. In gram-positive bacteria, porins are absent, whereas in gram-negative bacteria, porins are occur in outer membrane. Now, mesosome. In gram-positive bacteria, it is more prominent, whereas in gram-negative bacteria, it is less prominent. Now, morphology. In biological science, morphology means we study about size, color, structure. Now, in gram-positive bacteria, cocci or spore-forming broods are taken, whereas in gram-negative bacteria, non-spore-forming broods are taken. Now, flagella structure. Flagella structure means it is a thread-like structure. In gram-positive bacteria, two rings are found in basal body. This is basal body. In these two rings are found. This is called basal part. And in gram-negative bacteria, four rings are found in basal body. Now these four are basal part. Now lipid content. Lipid is a fluid substance. Now in gram-positive bacteria, lipid content are very low, whereas in gram-negative bacteria, it found in 20 to 30%. Now toxin. Toxin is a poison. In gram-positive bacteria, exotoxin are found, whereas in gram-negative bacteria, exotoxin and enterotoxin both are found. Now gram staining. In gram-positive bacteria, they appear purple color after gram staining. Example, Staphylococcus. Whereas in gram-negative bacteria, they appear pink or red color after gram staining. Example, Escherichia salmonella. Here, Escherichia means diarrhea and Salmonella means typhoid. Now, I will tell my friend to explain different application of gram staining.
Now I will explain you application of gram staining. Gram staining is essential procedure used in identification of bacteria and is frequently the only method required for studying the morphology.